Well, the video this week discusses various topics to do with blood, and the first of these are the components of blood. If you take a blood sample and put it in a test tube with an anticoagulant to stop it clotting, and then put it into a centrifuge and spin it, it separates out into different components. And the first is plasma, which is the yellow coloured fluid on the top. And plasma is made up of water, various salts or electrolytes, a lot of proteins are carried in the blood, and then there are nutrients, waste products, the respiratory gases oxygen and carbon dioxide, and hormones. Then if you look at the bottom part of our sample and test tube, the red component, you've got mainly red blood cells, which make up a bit under 45% of the total volume, and this is called a hematocrit. And it's a very important diagnostic tool when testing if a person has anemia. Then if we look at this narrow white layer, it's called a Buffy coat, B-U-F-F-Y, and it's made up of white blood cells or leukocytes and platelets. Now the next topic we go on to is haemostasis. So this is blood clotting and control of bleeding. This process is quite complex. It has three main phases. The first phase is where the vessel goes into spasm. It constricts because of a contraction of the smooth muscle in its wall, and that's to limit the amount of bleeding that occurs. The second phase is platelet plug formation, and platelets are attracted to the site and become attached to the collagen fibers in the wall of the blood vessel, and they collect together, and eventually a fibrin clot is formed to make a complete clot. Now this is quite a complex process, and the diagram below is just a summary of it. It's called a coagulation cascade. If you care to go to Wikipedia, and you can see that in detail. It's as I say, quite complex, but it ends up down the bottom with a protein called fibrinogen, which is soluble in plasma, being converted to fibrin, which then becomes the binding agent for the clot. Now this is a complex process because we don't want blood clotting occurring except under special circumstances, and you've been no doubt well aware of the problems with heart attacks and strokes, and these are both blood clotting issues.
Normally, blood clots form in response to injuries which cause bleeding. Clot formation helps to conserve the body's supply of blood so that small cuts don't end up draining the body of all of its blood. The body's process for stopping the bleeding by forming a clot is called haemostasis. In this illustration, the blood vessel contains red blood cells, white blood cells, platelets, and tiny proteins called clotting factors. These components of the blood float together in a straw-colored liquid called plasma. When the vessel wall is torn open by an injury, blood begins to escape. In response to this damage, the vessel quickly constricts, reducing the flow of blood. This constriction lasts only for a few minutes as the body puts in place a plug to stop the bleeding. Platelets soon recognize the injury and begin to stick to the damaged surfaces. This is called platelet adhesion. The adhering platelets change shape and release chemicals which keep the vessel constricted and draw more platelets into the damaged area. These platelets are said to be activated. Additional platelets arrive and begin to stick to one another so that a loose plug is formed. This is called platelet aggregation. When tissue is torn apart during an injury, blood is exposed to a chemical called tissue factor, which naturally resides on the surface of tissue cells, or may also be displayed on the inner lining of a blood vessel wall. This tissue factor sets into motion the chemical reactions of the clotting factors found floating in the nearby blood. Around a dozen different chemicals called clotting factors circulate inactively within the blood. When called upon, these clotting factors participate in a complex sequence of chemical reactions to ultimately produce tiny strands of a strong material called fibrin. Fibrin strands form in and around the loosely arranged plug of platelets and eventually form a meshwork which tightly binds the plug together. This meshwork keeps the platelets from being washed away by the flow of blood and makes the platelet plug more watertight. Red blood cells and white blood cells become trapped in the meshwork as it forms, giving the plug its characteristic red color. The overall process of forming these fibrin strands to solidify the clot is called coagulation. So within a matter of minutes, a sturdy plug is formed to arrest the outflow of blood, but too much coagulation activity allows the clot to grow much larger than it should. Two groups of the body's own naturally produced chemicals help to limit the size of the clot. The first group blocks coagulation. These substances are known as coagulation inhibitors or natural anticoagulants. When these chemicals interfere with the reactions among the clotting factors, fibrin formation is blocked, and no new fibrin strands are available to hold additional cells to the clot. In this way, clot growth is kept to a minimum. The other group of chemicals keeps clot growth under control by cutting the fibrin strands and actually dissolving part of the clot. These chemicals are part of the natural process known as fibrinolysis. Fibrinolysis occurs as chemicals called plasminogen activators are slowly released from the inner lining of the damaged vessel wall. These plasminogen activators trigger the destruction of the fibrin strands that hold the clot together. So coagulation inhibitors can only stop the growth of the clot, but fibrinolysis can actually dissolve part or all of the clot. In fact, when the vessel is completely healed, the remaining clot is dissolved by means of this second group of chemicals. To sum up then, rapid clot formation is essential for conservation of the body's supply of blood. We've seen the constriction of an injured blood vessel, the formation of a platelet plug, the strengthening of that plug by the addition of fibrin strands, and the body's protective systems preventing excessive clot growth.
Well, as you may have gathered, that wasn't my voiceover, but I found that an excellent explanation of the coagulation process. So our next topic is blood groups, and the main one we're concerned about is the ABO group. But first of all, some definitions. So antigens and antibodies are part of the body's natural defense mechanism. Antibodies are produced by the body in response to being exposed to an antigen. Now, commonly an antigen is something like a flu virus, for example, and antibodies are produced by the body against proteins in the cell wall of the virus. And when the antibody combines with the virus, it ruptures the cell wall. Now, in blood groups, you've got a similar system operating. It's explained a little bit more in this next table. This is the ABO blood group. It has 4 types. If people can be one of these types, either A, B, AB or O. Now, a person whose blood group A has anti-B antibodies in their plasma, even though they've never been exposed to B antibodies. And this makes this system different to a lot of other antibody-antigen reactions. A person whose blood group B has anti-A antibodies in their plasma. A person whose AB has no antibodies in their plasma. And a person whose blood group O has both anti-A and anti-B antibodies. This is explained in this next video.
And blood compatibility. Karl Landsteiner established the possibility of grouping blood into 4 types depending on their behavior when mixed together. Human blood has 2 major components, namely blood cells and blood plasma. The membrane of the RBC has 2 types of proteins called antigen A and antigen B. The plasma contains 2 types of proteins called antibody A and antibody B. The 4 types of blood groups are A, B. The red blood cell. That is, A group has antigen A, B group has antigen B. AB group has both the antigens A and B, while O group has no antigens in the red blood cells. The antibodies A and B in the plasma can react with corresponding antigens and coagulate the blood. So plasma of A group blood will have only B antibody. In B group blood, A antibody is present. AB group has no antibody, while O group has both A and B. The reaction between antigen and antibody has a lot of significance in medical science. Before any blood transfusion, doctors make sure that the blood groups of the persons involved are compatible. Let us see how this happens. Take 2 drops of blood from an individual with any blood group. To each of these drops of blood, add a drop of antibody A or antibody B. What do you expect to notice? If the blood drop coagulates with antibody A and not with B, it is group A. If the coagulation is with antibody B, it is group B blood. If coagulation is caused by both antibodies, it is group AB blood. And if there is no coagulation, it is O group blood. Conclusion. The blood group testing is based on the property that in any individual, the antigens and antibodies are never the same, because antibody A agglutinates antigen A. Similarly, the antibody B agglutinates antigen B.
Now we go on to another important blood group, which is the RH blood group. RH stands for the rhesus monkey, which is where it was first discovered. RH negative means that there's no RH antigen on the red cell wall, whereas RH positive indicates that the antigen is on the red cell wall. Now an RH negative person develops anti-RH antibodies only after they've been exposed to RH positive red blood cells. Now that's quite different to the reaction that occurs in the ABO system, as you've just learned. And at that stage nothing happens, and it's only when there's a second exposure to RH positive antigen that a severe reaction occurs. That's agglutination and breaking apart of the red cells. Now this is most important for an RH negative mother whose partner is RH positive. and so they'll be exposed through the baby to the Rh positive antigen in the first pregnancy. If there's a second pregnancy, then the severe reaction occurs, and so treatment has to be given to prevent this from occurring. The next topic is how gases are transported in the blood, and we can consider this at two levels: external respiration, which is at the lung level, and internal respiration, which is at the tissue level. So at the lung level, this is what happens. This can be shown graphically as follows. So you've got high concentration of oxygen in the alveoli and a low concentration in the hemoglobin in red cells. So you get movement of oxygen into the red cells, combines with hemoglobin to form oxyhemoglobin, and that's how it's transported in the blood. You've got a high concentration of carbon dioxide in plasma at this level and a low concentration in the alveoli. Now, carbon dioxide is carried as the bicarbonate ion, HCO3-, in the blood. At the alveolar level in the plasma, it's converted into carbonic acid and then quickly into carbon dioxide and water, and the carbon dioxide is offloaded into the alveoli. And this takes place under an enzyme called carbonic anhydrase, which speeds up this reaction enormously. Now, internal respiration takes place at the tissue level. And this can be shown visually in this diagram. We're at the tissue level, and here the carbon dioxide is at a greater concentration in the tissue cells, so it moves into the plasma where it's a lower concentration. It combines with water to form carbonic acid and then is quickly broken down into the hydrogen ion and the bicarbonate ion. And again, it's carbonic anhydrase that's the enzyme concerned. So it's the reverse of the reaction that it takes place in the lungs. And then at the in regard to oxygen, the oxygen level is highest in the red cell, so it moves out of the red cell to where it's low concentration in the tissue cells. Okay, that's the end of this video. Thank you for watching.